Hello, everyone, and thanks for the introduction. I'm Tatevik, a junior researcher at Armenian Bioinformatics Institute, but today I'll talk about a work I did at the Bigre Lab of the University of Oxford, where I was doing my master's degree. And I'll talk about SCRIMPy, our next flow pipeline for single-cell replication inference from multiome data using Python. So for those who might not know, the cell cycle is divided into these four main phases. G1, where the cells are growing and preparing for division. S phase, where they replicate their DNA. G2 phase, where they are continuing to grow and get ready for the division. And the M phase, where they actually physically divide. And it's important to study the cell cycle because it's a fundamental process which has impact on most of other cellular processes, such as differentiation, development, response to extracellular signaling, and importantly, response to treatment. And it's becoming increasingly clear that different cell types respond to treatment differently. And that's why the single-cell sequencing methods became very popular during the past decade. And for example, here you can see UMAP plots from a single-cell RNA-seq data, where you can see that different cell types in cancer have different distributions of cell cycle phases. And this distribution might change in response to treatment. And so it will be very helpful to be able to predict cell cycle phase of individual cells from single-cell data. So the existing tools for cell cycle phase prediction mainly use marker gene expression. So there are a list of genes that are generally highly expressed in a specific cell cycle phase. And these tools mainly use the expression values of such genes to classify the cells into specific cell cycle phases, as shown in this example heat map. So our lab was interested in gene regulation during the different phases of cell cycle. So we wanted to do an experiment of chromatin accessibility sequencing. And we decided to use 10x multiome sequencing because it not only chromatin accessibility data, but also gene expression data from the same single cells. But in order to do that, we need a golden standard method for tracking cells through the cell cycle. And that's why we use this genetic engineering approach called Fuji system. And I don't really have time to explain in detail, but this system essentially does is using a pair of fluorescent reporter proteins, a red one and a green one, which color the cells differently in different cell cycle phases. So as a result, the cells in G1 phase will get red color, those in S phase will get green color, and those in G2 phase will get orange color. So we use this genetic engineering method to genetically engineer mouse embryonic stem cells to have this Fuji system. And then we fluorescently assisted cell sorting to isolate populations of cells that are in G1, S, or G2 phase. We used these isolated cell populations and performed both single-cell RNA and multiome sequencing on those. And then we tried one of the existing marker gene-based cell cycle phase prediction methods from CIRAD package. And here you can see the results on the single-cell RNA-seq data, which are pretty good. With here you can see the isolated cell cycle phase populations on the x-axis and the percentage of cells assigned to a specific cell cycle phase here. And as you can see, the majority of cells in a specific cell cycle phase population were identified correctly. However, when we use the same tool to predict cell cycle phase from 10x multiome data, the accuracy was a bit lower, and this might be because the nature of the multiome sequencing when you isolate nuclei. And it's either because we don't really have enough information of gene expression in the nuclei, or the gene expression dynamics is actually slightly different in nuclei compared to whole cells. So here we had a question whether we can improve the multiome predictions by incorporating the single-cell ataxic data. And our hypothesis is that we can get information about the replication state of the cells using the single-cell ataxic data because, as we learned in this room already, there are specific regions, domains of the genome that replicate earlier and some regions that replicate later. And we think that more ataxic fragments are likely to be generated from early replicated regions compared to the late replicated regions in cells undergoing replication. And so this difference can indicate the replication state of the cell. For example, in G1 phase, when the replication is not started yet, we expect to see a uniform distribution of ataxic fragments in early and late replicating regions. However, in early S phase, when the replication starts, we expect to see more attack fragments coming from early replicating regions, as those regions will already have 2 copies of the DNA. And as the cell progresses through cell cycle, this difference decreases, and it becomes again a uniform, is expected to become again a uniform distribution during the G2 phase, when the entire genome is now already replicated. And we designed a metric called mean replication timing rank as a measure of this replication state. And here we used a matched replication timing data from mouse embryonic stem cells, showing how early each 50 kb bin of the mouse genome replicates. And we, from these replication timing values, we derive now replication timing rank with lower values showing earlier replicating bins and higher values showing later replicating bins of the genome. And then we simply calculate the number of ataxic fragments that are generated from those bins and the number of those with that generated from a specific replication timing rank. And then we get the mean rank for each cell. So for example, here, the cell 1 and cell 3 show relatively uniform distribution of the fragments across early and late replicating regions, while cell 2 has more fragments coming from the early replicating bins. And so we get, that's why we get lower value of the mean rank for cell 2, likely indicating that the cell undergoes replication, and so it is in S phase, while the cell 1 and cell 3 have higher replication rank values, and they might be either from G1 or G2 phase, where there is no replication in progress. So when we check the distribution of this metric across different phases, we actually see that for the S phase cells, we have lower mean rank values in general compared to G1 and G2 phase cells. And when using a threshold on this metric, we see that we are able to distinguish between the S and non-S phase cells with an accuracy of about 65%. Now, we know that different cell types have somewhat similar replication timing profiles. And in order to test whether we for our method, we actually require matched replication timing data from the same cell type or no, we tried to use replication timing data from another cell type, in this case, mouse neural progenitor cells. And again, we got a similar accuracy score. After that, we also try to infer the replication timing from the single cell ataxic data itself and use that inferred values for the for our method. And we see that, again, we reach similar or a bit more accuracy with that. So now we have one problem. With this metric, we were able to distinguish between S-phase cells from other phases, but we are not able to distinguish between G1 and G2 phase cells. And for that, we devised another metric, the percentage of overlapping fragments, as a measure of copy number to identify G1 and G2 phase cells. And here the idea is that if the cell has only one copy of the DNA, all fragments have to come from different regions of the DNA, just because of the nature of the sequencing protocol. But once the cells have replicated some regions of the DNA, then there is an opportunity to generate fragments which are actually overlapping and come from the same region of the DNA. And here I just highlight some examples of such overlapping fragments. And so we expect to get more overlapping fragments from G2 phase cells, as they have already replicated their genomes. And now we just count the number of overlapping fragments and derive the percentage of overlapping fragments for each cell as our next metric. And when we check the distribution of this metric across different cell cycle phase populations, we see that this metric can actually distinguish between G1 and G2 phases, and again, with about 65% accuracy. Then we wanted to check whether this overlapping fragments metric can also work on single-cell ataxic data from megakaryocytes data, as we know that megakaryocytes have a specific cell cycle and they can produce cell populations with different ploidy numbers. So we tested our method, and we, as a result, we saw that the distribution of percentage of overlapping fragments is actually quite different between the different ploidy groups of megakaryocytes. Then when we combine both of these metrics, mean rank on the x-axis of these plots and percentage of the overlapping fragments on the y-axis, we see that for G1 phase population, the majority of cells have higher mean rank. But they also have lower percentage of overlapping fragments. For the G2 phase, they again have higher mean rank, but now higher percentage of overlapping fragments. And for our S phase cell population, we see that majority has lower mean rank, but there are some fractions of cells, some proportion of cells, which show G1 or G2 like profiles, and this might just indicate the early and late S cells that are in early or late S phase. So then when we assign the cells to different phases based on our attack-derived metrics, and we compare our results with two marker gene-based methods, Surat and Tricycle, which were run on the gene expression part of our Multiome data. We can see that for specifically G1 phase cell population and S phase cell population, with our metric, we get higher proportion of cells identified correctly. But however, for the G2 phase cell population, our accuracy is lower. But as we generally have lower fraction of cells in G2 phase, it is better not to overclassify this phase. So now we showed that we can use single-cell ataxic data to infer replication state and to infer cell cycle phase. But for the future work, we plan to also incorporate the gene expression component of Multiome data to be able to get more accurate predictions, and also apply our method on to human data, as well as devise an automatic threshold detection method. and as well as try to test our method on other types of multiome data, such as single-cell histone modifications or single-cell methylation. So this is an ongoing work, and if you will have some suggestions, I'll be very glad to hear that. And with that, I want to thank my supervisor, Dr. Bigri, who is also in the audience, and all our lab members and collaborators for their contributions. And you can find our method, our pipeline GitHub repository with this link. And as a final note, I just want to use the opportunity to mention that I'm looking for a PhD position starting next year. So if you are interested, please let me know. And thank you so much for your attention.
Thank you for this very nice talk. So do I understand correctly that the Surat works nicely with single-cell RNA-seq, but single-cell nucleus doesn't work that nicely? But because we are talking about baselines today, the whole day, what would happen if you take this course of Surat and then just apply GMM, Gaussian mixture models, and then you say there are these three classes, what the accuracy would you get?
I'm not very sure. So you mean to incorporate both the Surat predictions with?
No, I'm saying that. So my understanding how this works, you have the two scores, and then if you one score like for G2M is high, you say this is G2M. But my understanding is that just thresholding doesn't work for single cell nucleus. And then I say, fine, so the thresholding doesn't work. But if you just do like you have these two values, two scores, and then you just apply a classifier, oh, sorry, not a classifier, a clustering into three classes. And then you say, now I have these three classes. Would it match your markers, your dyes, or wouldn't?
That's an interesting suggestion. So I don't think we tried something like that, but yeah, I think that's something we can think of trying for this data. Yeah, thanks so much for the suggestion.
All right. So first, thanks for the great talk, and perhaps this could be also helpful to study the complex interplay between the replication machinery and the transcription machinery, something you may want to look at. I have the question on the G2M. So there is also the possibility to have two cells in one droplet and to have doublet, and that may confound, that may be one confounder for you. Have you considered that?
So in terms of the doublets, we actually try to check whether we get, I think I also have we try to identify whether in our data we do have some doublets. So here I am kind of, you cannot, they cannot show. So here the coloring is by the doublet score, and for the, for example, for the G2 and S phase populations, there are some portions that have higher percentage of overlapping fragments, and they have a bit higher doublet score. So this might be also a problem, and this metric may even be helpful for doublet as well. Yeah, thanks so much.
We have an online question. Is there a reason behind the relatively less accurate prediction of G2?
I cannot think of a specific reason. No, that's, yeah, not really.
I have another question. You know, today earlier we were talking about these replication origins, and I may suggest a improvement or maybe additional metric, because if you have the replication origins, from them you would see your doubling feature that you see, right, which would start increasing. So instead of randomly maybe having these windows, there could be a way that you incorporate this information.
Mm-hmm. Yes, that's a good idea. Thank you so much.
Thank you very much.
Thanks.